Okay, so I'm going to switch from talking about the testing, which is what we've heard mostly about so far, to actually talk about targets, which is what I work on, and which is really why the Actionable Genome Consortium was formed. So the Actionable Genome Consortium was formed to try to fill in some of the annotation that Mia actually spoke about this morning, which is what are the drug targets, what do these things mean functionally, and how can we derive that data from some of the retrospective studies that have already been done, or even from some of the prospective studies like the ones that Roy mentioned. So really what my lab's been doing over the last 10 years and what I've been focusing on since I got into oncology was really to accelerate drug discovery. And I think it's a pretty simple way we try to do this. One is we try to define these targets, and I'll mention to you what the targets are and how we really classify these. We haven't heard much about it so far, but when we have a target, we need a drug because many of these paradigms or trials fail because we have poor drugs. And then when we have a drug and we have a target, what the real challenge has been over the last 10 or 15 years is identifying those patients who have alterations in a particular target, not necessarily so we can give them drugs as part of standard of care, so we can do clinical trials and test those things. So I'm just actually going to summarize what my thoughts are right here in the beginning and then provide some data. So if we have the correct patient and the correct target in that patient, why don't we see responses or cures more often? There's really four main reasons that drugs fail. One is that it's a bad drug, bad pharmacology. We haven't really heard about that at all today. So the drug just doesn't adequately inhibit the target. The second really relates to the fact that simply because the target's there, even if it's a good target like EGFR mutation, not all patients respond. And that's usually due to one of three things, either co-alterations, things that are co-mutated in the tumor that diminish addiction to your target. We also can have selective or adaptive resistance that I don't really have time to talk about today, but I do want to introduce the concept later of tumor subclonality and tumor as it relates to tumor heterogeneity and why that often leads to a lack of clinical response. So how do we define the targets? There's really two main approaches to this. One's called genotype to phenotype. The other's called phenotype to genotype. So quickly, genotype to phenotype is where we take a large cohort of tumors and in a retrospective fashion, we profile those tumors looking for things that are recurrently altered in the tumors. And this is really good to identify things that are common, things like BRAF mutation in melanoma, where it's 50%. And then when we have that mutation in BRAF, we know it's frequent. We think it's important because it's so recurrent and common. We develop a drug, in this case, vemurafenib, dabrafenib, take that into the clinic prospectively, test it, and ultimately end up with a drug. So that's one way to do this, and that works well with common tumors and things that are common within those tumors. But it doesn't work well with rare tumors, and it doesn't work well with infrequent mutations, even within common solid tumors. So there is an alternative approach called phenotype to genotype. This is where we take patients who have already responded to a drug, and we try to figure out why. We try to figure out what made that patient genetically unique. And one of the benefits to this approach is we already know that we have a good drug. So of those four reasons that trials fail, we've already passed the first one, which is at least in this single patient, the drug hit the target. We might not have known what the target was, but if we can figure that out, let's see if there's other patients just like this one. So we started an initiative called the Extraordinary Responder Initiative to try to take this approach. And we published some of these. So I'm just going to show one example that's unpublished because I think it's illustrative of some of the issues that have come up related to the testing today. So this is a patient with recurrent ovarian cancer enrolled on a clinical trial, GOG 239, of the MEK inhibitor from AstraZeneca, AZD6244. And as with all of the patients that we included in this initiative, this was a disappointing, relatively negative study. So this was the only patient who had a complete response. There were a few other partial responses, and overall the results were considered disappointing. But in this patient, who had actually failed 3 lines of systemic therapy and had no standard of care options at this point, she went on this trial back in 2009. She's now over 5 years out with a complete response. So this is a woman who should have died at that point within a short period of time of her cancer, given a drug that otherwise failed in most patients that was disappointing and has had a complete response. So what made her unique? So we performed retrospective next-gen sequencing to analyze her tumor, and it turns out that what she has is a 15 base pair in-frame deletion of a gene called MAP2K1. And so what is MAP2K1? Well, MAP2K1 is the gene that encodes MEK1, which is the target of the drug. So again, it doesn't have to be complicated. We simply didn't know that this patient had a MEK1 mutation going into the study. And even if we knew that prospectively, we didn't know what this specific alteration meant. But seeing this patient have this incredible response really points you into the direction of saying this is probably a functional alteration and it's probably important. The other thing I want to point out from this patient is that this is a tumor, low-grade serous borderline ovarian cancer, which is really characterized by a lot of stromal infiltration. And there's no way you're ever going to get one of these tumors with greater than 70% tumor content. And you can see only 8.3% of the alleles actually had this alteration, but we easily were able to detect it. So this issue of having perfect tissue is not necessarily always a requirement. It's definitely, to me, not a requirement in this day and age of deep next-gen sequencing. So what does this mutation mean? You always have to go back to the lab and functionally validate these things. If you knock out the 5 base pairs that are encoded by this deletion, you actually knock out part of the negative regulatory helix here. And if you go ahead and you make this mutation in the laboratory, you find that it locks the MEK1 protein into a constitutively active conformation that the MEK inhibitor is able to inhibit. So in retrospect, we're able to identify why this patient responded and actually learn a little bit of biology that if you delete this part of MEK1, that actually activates the protein. And then can we use that information for the next patient who comes along with a similar type of mutation? And that actually happens. This is a patient with melanoma who was part of our next-gen sequencing prospective efforts. And I just blew this up for people who can't see it. This patient has a MAP2K1 mutation, actually has a deletion that involves 2 amino acids, so 6 base pair deletion here. It's an LE101, 102, so you delete 2 other amino acids a little bit further down the protein. And the question is this an activating mutation? So if you go ahead and you make this mutation in the laboratory, here's this mutation right here, you get very high activation of phosphoric, similar to what you see with that mutation I just showed you in the extraordinary responder, similar to the K57N mutation, which is the most common MEK1 mutation in lung cancer. And even there, only found in about 0.6% of patients with lung cancer. So the question is, we can find these alterations, we can even find them prospectively. Is there any way to get the MEK inhibitor for this patient so we can test that simple hypothesis? If you have this MEK1 mutation, do you respond to a MEK inhibitor? So how are we going to do this? I'm going to glaze through some of these slides because you've seen these concepts. So one way to do it is to have a master protocol like Roy mentioned, or an umbrella protocol, or you could call this a match protocol. I actually don't consider the NCI study to be a basket study. It's more an umbrella, or even you heard it from Lillian, a tent protocol. And here you genotype all the patients from a particular cancer, and then you allocate them essentially to different drugs depending upon what type of mutation you have. And there's a few problems with this approach. And the problems are that the drugs are not always best in class, but what's available to the investigators at the time that you design the study. So this is a major problem with these studies. You have to negotiate with multiple companies to bring these drugs into your umbrella, and you may not have the best in class. 2, if you actually do an adaptive randomization, and this was true on the initial battle study, it can actually become unethical during the course of the study to randomize patients to some of the arms. So personally, if I was seeing that, for example, there was a 42% response with brafenib in a patient with BRAF lung, and I was on an umbrella study that's going to adaptively randomize me to an EGFR inhibitor, I would pull out of that study and I would go, try to get the BRAF inhibitor. And then the key point I really want to make is that the total number of patients is generally too low on these studies to identify sufficient patients with rare alterations in rare cancers to actually ask your primary question, that genotype to phenotype question. So what's the alternative? The alternative that we've been pushing are these basket studies. And so again, instead of designing the study around a disease, you design the study around a mutation. This was the design of the initial BRAF basket study led by Jose Baselga, led at our institution by David Hyman, where it doesn't matter what kind of cancer you have, as long as it wasn't melanoma for this trial, and you didn't have to put melanoma patients on the study because the drug was already approved in melanoma, so there was no point in putting them on. But all other cancers were eligible, and then statistically, it's a very simple design. It's just a bunch of parallel phase 2 studies, and what we've learned is that it doesn't work in colorectal cancer as a single agent, 42% response rate in lung cancer. I don't think we found any patients with breast cancer. Erdheim-Chester, it's about 100%. So it's different in different cancers, and that's what you can learn from these type of trials. And I just want to highlight this patient because of some of the questions that were asked this morning. This is a patient with Erdheim-Chester disease. This is non-Langerhans histiocytosis. This is a disease that we didn't even know 10 years ago was a cancer. We thought maybe it was an autoimmune disease, and it turns out that it is a cancer. These are all clonal. It's a clonal infiltration of these histiocytes, and about 50% of these patients have BRAF V600E mutations. And I would actually point out, it was just published a few months ago that 25% of these patients have these deletion mutants in MEK1. So that hasn't been clinically tested, but probably is going to work really well. This is a patient who actually had infiltration of her cerebellum. So she had brain infiltration with the disease. She was wheelchair bound when she started on the trial. She's had a complete response. This is over 2 years. And it's back to her activities of daily living. And the reason I really wanted to point this patient out, not just because she's had this extraordinary response, but to actually point out that we actually tried to bill her insurance company for the BRAF V600E testing that we did for this patient, and they actually rejected it. Even though she had a complete response, they said, you know, BRAF testing's not standard of care in Erdheim-Chester disease, and we're not going to pay for this. And so what happened, well, actually I paid for this through actually using my own laboratory funds, you know, which is supporting some of this. So, you know, how many R01 investigators take some of their laboratory funds and pay for clinical genomic testing? But this really just had to get done. So this is another trial. We've gone out and looked for other studies where this would be a great paradigm. This is a study I lead. It's a study of the HER2 or pan-HER kinase inhibitor neratinib. Again, a basket study. You can have a HER2 mutation in any cancer. You can have a HER3 mutation, any solid tumor, and then we have an arm for EGFR mutations in the extracellular domain if you have a primary brain tumor. And one thing to point out about these studies is it doesn't matter how many prior therapies you've had. As long as you have a mutation, you're eligible for this study. So even in Roy's squamous lung cancer patients, if they've had more than one prior therapy, they're eligible for this study. We don't put any limits on prior therapy. So they are somewhat complementary to the master protocols that you've heard about, which are sometimes a little bit more restricted. So does it work? Well, this is one example. This is a patient with breast cancer. So she's considered HER2 normal because she doesn't have HER2 gene amplification, but she has a V77L mutation. This is a mutation in the kinase domain of HER2 that causes a HER2 activation similar to an L858R EGFR mutation. And you can see all of this disease that she had pre-treatment. 2 months on to the therapy, she's had a complete response. It's not the only patient we've had with a response like this. And what I would really try to highlight is that essentially almost all patients with this mutation are unaware that they have this mutation because we don't test for this mutation. So almost 98%, I would say, of the patients with this mutation have no knowledge of that fact. And that's despite the fact that we have this incredible response. So what we're really talking about here over the last day or so, and what people are going to talk about tomorrow, is how can you not be testing for these type of mutations in diseases like breast cancer? So what are the advantages of the approach? It allows us to test a defined biologic hypothesis. I see patients, but I also run a research lab. We want to ask the question, the simple scientific question: if you've got a HER2 mutation, do you respond? And I would point out that to actually do this study, and actually was highlighted by Anne-Marie on the last talk, we've estimated that between 30 and 40,000 patients are going to need to be screened to do this study. And we've been given absolutely no money from the company to do the screening. So we're going to accomplish this study with absolutely zero pharmaceutical support for the screening. And the way we're going to do that is we're going to get other people to pay for the screening, okay? And we're going to do that by getting people at MD Anderson to screen their patients. We're screening our patients. Foundation Medicine's screening 20, 25,000 patients a year. All we need to do is let them find those patients for us, and then just allow the patients to go on the drug by having a study that's available for them to be enrolled on the clinical trial. So we can ask this simple question. We can ask, does lineage matter? Does the specific mutation matter? We can collect tissue prospectively, and then look retrospectively for tumor heterogeneity, and then we can do this co-clinical trial. So what we do on this study is we allow patients with any HER2 mutation to go on the study. It may be a known activating mutation. It may be an uncharacterized allele. And when the patient goes on, we actually make the mutation, as I showed you with MAP2K1, in the lab and we ask, is it an activating mutation or not an activating mutation? And in some cases, we may find, it'll probably take us 4 months to do the experiment, but we find that it wasn't an activating mutation and maybe the patient didn't respond. And did we harm the patient in any way by exposing them to that treatment? I don't think so. I would go back to this patient and point out that she's had 4 prior therapies, none of which she responded to before getting this drug, and then went on to respond to this treatment. There is no standard of care for this kind of patient. So I think putting that patient on to the trial, I think, was reasonable. But we may have put on patients who have other different mutations, and this is just showing the diversity of HER2 mutations in cancer. And it's very possible that this patient, that these type of mutations respond less well than the ones here in the kinase domain. We simply don't know that. We're going to learn it from the trial, but we're not excluding patients due to our lack of knowledge on the topic right now. The other thing that we've talked about, and we're getting ready to already do this in breast cancer, is we start out with a basket in patients with breast cancer. We enroll 18 patients with breast cancer with HER2 mutations onto the study, but they have different alleles. And it may be that we need to actually narrow it down further to figure out what's the response in a particular mutant subtype. So we can actually take the breast basket, and we can actually divide it up to multiple baskets, a basket just for breast cancer patients with S310, breast cancer patients with L755, breast cancer patients with B77L, eventually get sufficient numbers for each of these different mutants to ask the question, what's the response rate of the drug in that particular mutant drug combination? And could we ultimately find a mutation-disease combination where the response rate and durability response is sufficiently high to warrant an immediate change in clinical practice? Because I would point out there's probably only 100 patients in the entire country a year who have breast cancer and this particular mutation at most. There might even be less than that. So there's no way you're going to ever do randomized clinical trials in these distinct subpopulations. What are the challenges with this approach? The primary criticism we get from clinicians, companies, regulators is that we may fail to identify patients who may potentially respond, but they lack the biomarker being tested. It's completely true. We're only looking to see if you've got this mutation, do you respond to the drug? It's possible there's other mechanisms to respond to the same drug. That's true, but it's not what we're testing in this particular trial. We can try to explore those things in the future. I call it the sad fact, and this is getting multiple disease teams to work together has been a challenge with these basket studies. And it gets down to the breast group wants their own study, and the lung group wants their own study, colon group wants their own study. And that usually works out for lung and for breast. Doesn't work out great for patients with Erdheim-Chester disease because there isn't a lot of those patients who are advocating for clinical trials in Erdheim-Chester. But even diseases like bladder cancer, which are not that common, these basket studies provide sometimes an opportunity for these target-directed studies for the first time because many major pharmaceutical companies haven't been particularly interested in these rare cancers. What's the primary hurdle? Well, you've heard about it all day. Identifying the patients remains a challenge. If we're not screening for the mutations, we can't find them for the study. So it's at least my belief, and this is totally my opinion, that the screening protocol should be separated from the treatment protocol. This is very polarizing because it gets down to who's going to pay for the screening. The drug company doesn't want to pay for it if it's not part of the therapeutic protocol. So how are you going to screen for these patients? So the way we're doing it at Sloan-Kettering is we've developed our own custom in-house assay, and we've been very cognizant of the cost of this assay. We've tried to do this by driving down the price of testing so that we can screen a large number of patients. And our current assay screens 341 cancer-associated genes, and this is going to jump up to about 410 in the next few months using a capture-based approach, similar to what Foundation Medicine is doing right now. You've heard this a little bit as well. We take all the data from the clinical profiling and we make it available to every single investigator at Sloan-Kettering in real time. So we use the CBio portal that many people may be familiar with to mine the clinical data. I actually took this screenshot last night. It gives an immediate update of how many patients we've screened. We've screened 1,768 tumors from 1,727 patients. 304 breast cancers, 211 lung cancers, all the way down the line, 62 bladder cancers. You can click on these. You can see the profiles of the patients that we've screened. You can actually look through each individual patient's mutational profiles. And so what this does by screening so many patients is it makes finding rare things relatively interesting. This is actually, again, a screenshot I made last night of actually a partial list of the HER2 mutations we've already found in the last couple months. We only got New York State approval for the assay in July, conditional approval in late April, early March. And you can see we've already found a lot of patients with HER2 mutations. We've actually found 8 patients already with S310F mutations. We were told this clinical trial was not accruable. Again, once you start screening every single patient for the mutations, finding things that are even rare become relatively easy. We've already found 4 patients with L755 mutations. 3 of those are breast, 1's endometrial, mostly bladder with S310F. That's something we're seeing, that which mutation in HER2 you find actually does correlate in some ways with what disease the patients, in fact, have. So I just want to show you just a couple cases, which, again, point to me that it's actually the biology that's going to be the difficult part here, not the testing. We've talked a lot about testing, but that's probably not going to be the main issue. This is a patient with a HER2 mutation, but they have a KRAS mutation. And for those aware of the data in this setting, in lung cancer and others, in all likelihood, having a KRAS mutation is going to make this patient resistant to a HER2 mutation. But we actually don't formally know that. So the question is, would you put this patient onto the clinical trial? Here's a patient with a HER2 mutation that would likely respond. It's actually clonal. But the next patient here has a HER2 mutation, also has a TSC1 mutation. But the reason I'm showing this HER2 mutation is that the mutation's only found in 3% of the sequencing reads versus these other mutations where you see it found in the high 30s or 60s. And that means that this HER2 mutation is probably a subclonal event. It means that only a subset of the cancer cells likely have this mutation. And so is this patient likely to respond to the treatment? So to me, it's not an issue of sensitivity of the assay. It's the biologic data that underlies the cancer that's probably going to maximally impact our patients and what we should probably be focusing on. So what is the Actionable Genome Consortium going to do? It's a combination of the NCI, Memorial, MD Anderson, Broad, Cancer Research UK, et cetera, many other institutions. And what we're really going to do is try to demonstrate clinical utility, democratize genomic testing by making it more widely available, try to drive down the costs of doing the testing, define actionability across these institutions, and all the conclusions are going to be published and made available to the community without any restrictions. And ultimately, again, we're going to define a suite of standards, many of the things that you've already heard about today, so I'm not going to revisit at this point, and I'm just going to leave it there. Thank you.
Great. Thanks, David. Next, I'm going to invite Sean Tunis to come up from CMTP, and Sean's going to talk a little bit about a concept that we've had mentioned several times already today, which is coverage with evidence development.